Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, my name is Nurlaila Sari. I'm a student majoring in Food Science and Technology, Hasanuddin University of Makassar. Today I will present my paper entitled Isolation and Identification Lactic Acid Bacteria of Honey Enriched Functional Beverage from Cassava Tape from Sinjai Regency. Introduction. Tapai is fermented cassava product. Tapai main ingredient of the functional beverage that contain lactic acid bacteria. Next, honey. Honey is one of the ingredient at the manufacture of the functional beverage formula. And based on the background, this study aimed to identify lactic acid bacteria found in the functional beverage based on cassava. Next method, the tools that are in form of HPLC, laminar airflow, autoclave, oven, incubator, water bath, microscope and glass object, DNA thermal cycle, electrophoresis and type supply, UV lamp device, and digital camera. And the material used in functional beverage of cassava tapai, Indonesian organic honey, MRS agar, MRS broth, Aquades, NaCl, cotton sterile gauze, aluminium foil, violet crystal, lugol, ethanol, safranin, media SIM, liquid media MRVP, TSIA media, oxbile salt, hydrogen peroxide, and hydrochloric acid. Next, table for organic honey analysis result. Water component of honey is 21.5%, fructose component 35.2%. Next, glucose component is 24.9%, but sucrose is 0%, and the last pH 4.2. The result of characterization in table 2 is that the gram coloration of 19 isolate in the form of basil and 1 isolate of coccus, the red color, which means that the isolate gram negative. Otherwise, if the visible color is purple, mean the isolate include gram positive. Next, the TSIA test show 16 media that change color to yellow, red, and 4 media change color to yellow. This indicates that the media with red slant part and yellow bud mean that the bacteria are able to ferment glucose, while the media of the slant and bud are bud yellow, meaning that the bacteria are able to ferment sucrose and drugs. And out of the 20 isolate are grown in acidic condition. In the bile salt test show 15 out of the 20 isolate grow in the condition. In general, that are 2 isolate of bacteria growing well on the resistant test against temperature, acidity, and bile salt that are M3D and M5B isolate code. Both isolate were subsequently conducted molecular tests for the indication. Both samples were M3D and M5B were amplified using the primary 63F and 1387R and show a thick and bright ribbon. It demonstrates the sample DNA has a high concentration and indicate amplification using primary pair of 63F and 1387R. Discussion. Pediococcus acidilactis is a gram-positive species often found in fair or tetrads. Pediococcus acidilactis is a homofermentative bacteria that can grow in a wide range of pH, temperature, and osmotic pressure, thereby being able to colonize the gastrointestinal tracts. This species has emerged as a potential probiotic that has demonstrated promising results in animal and human experiments, although some results are limited. It is commonly found in fermented vegetable, fermented dairy product, and meats. Pediococcus is an antagonist towards other microorganisms, including enteric pathogens, mainly through the production of lactic acid and the secretion of bacteriocin known as pediococcins. For the Wisella species have been isolated from various sources, and some of them play an important role in fermentation. Wisella sibaria was first described by Brockart et al. And then found in various types in fermented food. The conclusion, based on the result of research that has been done on the functional beverage of cassava tape with the addition of honey treatment, can be concluded as follows. Number one, honey added to functional beverage of cassava tape is honey with water content and sugar sucrose in accordance with SNI or Standard Nasional Indonesia. Number two, the result of identification lactic acid bacteria with the potential probiotic, namely Pediococcus acidilactis and Wisella sibaria. Okay, thanks for the change and see you next time. Assalamualaikum warahmatullahi wabarakatuh.